Today we'll be talking about population genetics and how certain pressures can impact the composition of a population in question. To demonstrate this, we'll be using straws, a diploid organism at 2n equals 10. The autosomal gene in question will be color, whose two alleles are O and P. There are three genotypes OO, OP, and PP, which correspond with three phenotypes of orange, green, and pink, respectively. First, I want to introduce the Hardy-Weinberg law, which states if certain assumptions are made for genetics of an observed population, the frequencies of alleles and genotypes will reach and remain at equilibrium after one generation of random mating. All this is saying that if there are no internal or external pressures placed on a population, their genetic makeup remains the same. The assumptions mentioned are infinitely large population, random mating, no selection, meaning there is an equal fitness amongst all genotypes, no migration, no mutation. We will focus on no selection today. Back to our straws. Here's our population of various straws within Straw Nation that we will examine. 24 orange, 48 green, 28 pink straws for a total of 100. To determine if this population is in Hardy-Weinberg equilibrium, we need to calculate the frequencies of O and P, or alleles which correspond to P and Q. Are there 100 straws with two alleles each? We'll divide the total number of O and P by 200 to get 0.48 and 0.52 for our frequencies. Now that we have P and Q, we can solve for P squared, 2PQ, and P squared, which will give us our values of expected genotype frequencies, meaning how often we should expect each of the three colors. Next, we'll perform a chi-squared test using all the values we have calculated. For the scenario, our null hypothesis will be our population is in Hardy-Weinberg equilibrium. To get the number of expected, we will multiply our expected genotype frequencies by 100, our total number of straws. We get a chi-square value of 0.15. As our degrees of freedom is 1, 3 genotypes minus 2 alleles, we will compare 0.15 to our critical value of 3.84. As our critical value is greater, we will fail to reject our null. We can conclude that at these parameters, the color of straws is consistent with Hardy-Weinberg. As previously mentioned, selection can violate the Hardy-Weinberg law. For example, our respective fitness values are 1, 1.2, and 0.8. To calculate average fitness, we'll take a weighted average of our fitness values. Our adjusted survivor numbers are as follows. To determine the new genotypic frequencies following the selection, we divide the unadjusted survivors by the average fitness. This is significant as you can see that orange went from 0.24 to 0.23, green went from 0.48 to 0.55, and pink went from 0.28 to 0.22. Now let's look back at Straw Nation with our new frequencies after selection. There's 1 less orange, 7 more green, and 6 less pink. The type of selection seen here is called stabilizing or overdominance. In this mode, heterozygous straws green are favored. Pink and orange are selected against, but both O and P alleles remain in the population. This makes sense as Straw Nation is uniformly green, allowing for the green straws to blend in better and avoid predators. If this form of selection continues, the frequency of greens will continue to increase in a gradual fashion, as seen by this graph. This drive towards the heterozygous reflects how Hardy-Weinberg is being violated due to selection pressures and differences of fitness.